Well, first I want to say thank you to you all for the opportunity to come speak to you today about some of the work that is going on in my group and some variations of the theme that I think apply very well to the topics of this institute. And so the title is meant to be a little bit broad, the idea of engineering cellular microenvironments and microstructures. The concept behind that is sort of twofold. My lab works in fabricating scaffold type materials, which are porous biomaterial structures, and one of the ways in which they can be described is as cellular materials. And the flip side of the title is that we actually use these materials not only for sort of traditional regenerative medicine purposes, but also as model systems to study cell behaviors. So that's sort of the duality of where the title came from. And so this stems out of the sort of a generic slide that people present a lot, is the idea of the promise of tissue engineering. And for a long time, tissue engineers have worked to bring together different areas of research, mainly biomaterials, soluble factors, and cells in some combination. And the main goal has been to repair, replace, restore, regenerate some lost tissue. And there's been lots of areas in the body that people have worked on. You know, musculoskeletal is a classic example. There's a lot of connective tissues that are becoming more and more interesting and important. Cardiovascular systems are extraordinarily important. Nervous system, lymph and immune system. These are all different types of tissues where people have been trying to develop biomaterials. And what we've learned along the way is that maybe the field of tissue engineering is not necessarily going to be growing an entire liver in the lab and implanting it into a patient. Maybe we have to think about a lot of different ways in which we can actually use the tools of tissue engineering to sort of spur what hopefully is going to be the next generation of advanced materials that we can use for biological applications. And so where the field of tissue engineering is starting to move is away from just making material to put in the body or trying to grow a whole tissue outside of the body, but how can you develop material systems that allow you to identify pathologies more rapidly? So instead of having to replace a whole liver, if you could diagnose liver disease early enough to be able to treat it, you won't get to that problem. Similarly, is there ways in which you can use model systems to quantitatively probe from the complexities you see in biological systems, and just in general develop new tools that biology medicine can use for a whole host of applications. And so a lot of this is based out of, and my work is based out of the concept of the extracellular matrix, which is it's the insoluble regulator cell behavior. It's a complex organization of structural proteins such as collagens and proteoglycans that you find in all your tissues and organs in the body. Each tissue is a slightly different type of extracellular matrix structure. It could be differences in composition, mechanical properties, microstructure. There's a whole host of ways in which these can change. And what we found as tissue engineers is as we started developing analogs of this extracellular matrix for tissue engineering applications, some examples of which have been scaffolds and gels, that there's a lot of critical characteristics that need to be optimized within these extracellular matrix analog structures. Some of the main ones that my group and lots of other groups around the world have been working on, so are looking at ways in which you can change chemical composition and degradation kinetics, microstructural organization, so the porous nature, the size of the pores, the shape of the pores, the amount of solid versus porous structure within the material, mechanical properties, different biomolecules, whether they're being soluble release of biomolecules or insoluble tethering of biomolecules to structures. All these things have been found to be very significant in affecting cell biology, but what, as a field, we've been trying to develop is ways in which we can develop more of a quantitative understanding of how these different properties of a biomaterial structure can affect cell biology. So my group is sort of a broad-ranging group here at the University of Illinois. My background's in material science and mechanical engineering, actually. And our group works in fabricating collagen-based biomaterials, mainly scaffolds, these porous three-dimensional structures. I liken them akin to sort of a kitchen sponge, as opposed to a hydrogel, which is closer to Jello, for example. Although we do a little work with hydrogels, most of our work is focused on developing these three-dimensional porous scaffold structures. And we use these for a whole host of different applications. So we're basically a biomaterials lab that looks for applications and try to work out ways that we can develop our systems. One of the big areas that we've had some success in is generating biomaterials for orthopedic tissue engineering applications, both in vivo and in vitro. We've also used these biomaterials as model systems to study the effect of extracellular cues on cell behavior. And we've been starting to work with ways in which we can develop quantitative modeling tools to describe our biomaterials. So we can do experimental work, we can do theoretical work as well, as ways in which we can describe our materials. And a lot of this is actually building up to ways in which we can develop systems to study stem cell behavior. I'm not going to talk as much about this today, only because this work hasn't really transitioned into using some of these cellular biomaterials quite as much, although hopefully in future years that some of that will be a little bit more to the forefront. So let's talk about how we make these biomaterials. So I said before, these are collagen-glycosaminoglycan, or CG scaffolds, and they're fabricated by making an aqueous suspension of our different proteins. So in this case, type 1 collagen and chondroitin 6-sulfate is sort of our standard material we work with. We mix them together to form this aqueous suspension, and then we solidify it. And so the way freeze drying works is it actually utilizes the concept, the idea where you take your suspension in the liquid state, you freeze it to get an interpenetrating network of ice crystals surrounded by the collagen and glycosaminoglycan content. And then by pulling along that vacuum, you can actually sublimate out the ice crystals to form a pore structure. So the concept is that wherever you have an ice crystal that forms during solidification ends up being a pore in your final scaffold. So by changing features around the solidification kinetics in terms of the speed or the temperature or the directionality of solidification, you can actually change the size and the shape of the pores in quite a rigorous manner. And so what we've been able to show is that, as you'd expect for a lot of solidification kinetics, you start out with this process where this is a thermocouple put inside one of our, inside the suspension as it's freezing, is you start out with this undercooling phase, and then you have an ice crystal nucleation phase. Then you have this coarsening phase, which is what actually defines the size and shape of the ice crystals. So controlling the thermal treatment at this phase is where we actually do a lot of our work to change the size and shape of the final scaffold structure. And then there's a cooling phase. And what you end up with at the end is these nice low-density open cell foams. Most of these scaffolds are anywhere between 0.5 to 5% dense. So that means their porosity is between 95 and 99.5% porous. So these are extraordinarily porous materials. And what you're seeing here is an SEM image of one of our scaffolds, and the white content is actually the collagen glycosaminoglycan structure, and the dark regions are pores. And you're actually seeing many pores into the structure via this SEM image. And so there's lots of other ways in which you could generate similar cellular materials. I didn't want to give the suggestion that freeze drying is the only way you could do it, so I decided to put up a couple of other examples of groups that have developed similar cellular materials. One of the classic examples is using colloidal crystal templating, where you take some spherical templates, sonicate them together to form a packed structure, and then fill in the space in between with some biomaterial, and then grade out the spheres. And what you end up with is a porous structure that is a little bit more regular than our collagen gang scaffold. But again, in looking at a confocal microscope image through it, you can see these nice regular pore structure where you have a great deal of open pore nature, some solid, and interconnections between the pores. Other templating approaches have generated similar materials, although not nearly as uniform. Some of those examples are salt leaching and gas filtering. So why am I sort of giving you this idea why less people do hydrogel type work? We happen to do a lot with scaffolds, and we feel like there's a number of ways in which scaffold type materials actually give us some advantages over hydrogels. One of the big ones is that because of its low density open pore nature, we can actually make the scaffold, let it sit for a while in a dry state, and then go through hydration and seeding and implantation. So we don't have to get our cells into it like in a hydrogel where you get cells into a hydrogel instantaneously upon formation. Our cells actually have, could you guys please turn off your cell phones? I mean, seriously. Okay, thank you. Anyway, so when you get these cellular structures, we actually have some possibilities to do some modeling work, and that's one of the advantages that we bring to this. So I'm going to talk a little bit about this concept. How do you model cellular structures? So this applies very well for our scaffolds, applies to a lot of natural materials and other biomaterials as well. So what is a cellular material? Well, as you've sort of gotten a sense from our scaffold structures, these cellular materials are low-density, high-porosity materials. And so there's lots of examples. There's structural, in this case, aluminum honeycombs that are used in aircraft parts. There's lots of open and closed cell polymeric foams that are used for, again, structural applications, bumpers, and those types of materials. But there's lots of cellular materials found in nature and medicine as well. One example is trabecular bone over here. Trabecular bone is a very low-density open foam structure, and similar with our scaffolds. Again, the consistency between these is that each one of these materials may have a slightly different structure, but the concept is the same. You have high porous materials that have interconnections between the pores within the material. And there's actually a modeling framework that one can use to start to think about it. So one of the things that we care about as biomaterials is mechanical properties. And so that sort of started us thinking about using modeling approaches, but it actually has expanded to much farther than that. So what we started to use is this concept of cellular solids modeling. And so what this means is that we can take our experimentally produced biomaterials and actually put together a modeling framework to describe aspects of these materials. And so this comes out of the idea that the relationship between microstructural elements in three-dimensional porous materials is actually quite complex. It's difficult to model exactly, but you can use dimensional arguments. So you could actually take our three-dimensional scaffolds or trabecular bone or some other polymeric materials that are used for structural applications and describe them using a unit cell concept where you describe the average pore in terms of this is a square prism unit cell, there's a tetrachydecahedron, which is a 14-sided unit cell. There's lots of different unit cells you can use. There's some slight variations between them, but what we found is that the dimensional scaling arguments you can make using these types of materials actually allow you to make a lot of predictions about how your materials behave. And that allows you to start to make connections between macroscopic properties you can measure and microscopic features that, for example, a cell might be exposed to when they're inside materials. So what does that mean? So how do these materials behave? So let's take a classic example. If you take any of our porous materials and you compress them and try to get a sense of what their mechanical properties are, they have very much a conserved series of properties associated with them. And what you see when you take your biomaterial or your porous material is you can put together a stress strain curve under compression. What you see is an initial linear elastic region. And this is as you start compressing your material, all the individual fibers within the 3D structure start to bend. Then there's an elastic buckling stage, which can go from about 10 to 60 to 80% strain. These materials can be compressed down to 95+% strain because they're low density in nature. This elastic buckling plateau is associated with all the fibers within the material starting to buckle and collapse on top of themselves. And then you get a densification event that occurs when the fibers are compressed on top of themselves, and then you're actually compressing fibers on top of fibers. And so this is sort of a generalized one to look at what you might expect to see for the generic low density open cell bones. We've seen this behave quite well for a whole host of different structural materials. These are elastoplastic materials that are low density open cell bones. Depending on the density, you get different behaviors, but fundamentally, this takes the behavior is somewhat similar. And we even see this in our collagen gag scaffolds. So this is compression up to about 95% strain, and what we see is this nice linear elastic plateau and densification regions with very distinct planes that we can plot to describe mechanical properties of features. All right, so why might you care about that? Well, let's go back to thinking about this idea of dimensional analysis and dimensional arguments that you can make. I'm going to do a lot of this using the square prism unit cell only because the math is is the easiest of all the different cellular materials, although there's very similar types of work you can do for more complex cellular structures. So let's start thinking about ways in which you can describe the modulus of a cellular material, and this using this type of modeling framework. So when you think about using a dimensional argument, you have an individual unit cell that has given associated lengths to the fibers and associated thickness of each fiber. So if you start thinking about trying to describe the relationship between the applied force on this unit cell and the stress in the unit cell, which scale is a length squared, which would be sort of the top surface of this biomaterial, and there's also a relationship between deformation and strain, again by length. So then you can start describing what the deformation of one of these fibers might be under load, and actually is a column buckling equation you can use. And by taking together, and the last feature you need to start to define is this idea of relative density, which we're going to talk a lot about in the course of the rest of the lecture. The idea behind relative density is that this is a measure of how much solid is in the material. So if you think about calculating a relative density for this square unit cell, what you would do is you take the total volume of material, which would go as length times thickness squared times some constant, and divide it by the total volume of this unit cell, which would be a length cubed sort of relationship. And so that gives you a relationship. Again, this is not a direct relationship, but it's a proportionality that the relative density of material is proportional to the ratio of thickness to length of the individual fibers that describe the material. So you can start to model this together, and when you combine the buckling equation with your sense of what stress and strain is within the material and the relative density, you can actually come up with a relationship between the mechanical properties of the porous foam itself and the mechanical properties of the fiber which the foam is made out of. So that's something that we might, as material scientists, be very interested in understanding. And it scales exactly as the relative density squared. So that means if you have a material that is 1% dense, and you double its density to 2% dense, you expect the modulus to quadruple, for example, because it's the relative density squared. So this is actually conserved quite a bit in terms of looking at Young's modulus of a whole host of different bones and different relationships, and a whole host of these are metallic biomaterials, porous biomaterials like porous foams, porous metal foams used in hip joints and the like, and it's quite conserved across a whole host of porous biomaterials. So how does this now, so now to give you an idea of what cellular solids modeling might be, how does this actually apply to what we do? And so what we just try to do is we try to create a homologous series of biomaterials. And the concept behind homologous series of biomaterials is that you want to take one biomaterial and make a number of variants of it where you're changing only one property, and you're keeping everything else constant. And the idea behind why that is important is, especially as you start to try to make quantitative measurements of cell behaviors, or if you're trying to optimize a material for a particular application, it's very useful to know exactly what the structure-function relationships are that you're measuring. And so we have ways in which we've produced a number of different homologous series of our scaffolds, one of which is by changing the freezing temperature. This goes back to the fabrication method I talked about. We can change the pore size of the scaffold without changing its relative density, because we start with a constant suspension, has the same amount of collagen in all the suspensions, and so what you get out is a three-dimensional material that has constant relative density, different pore structures. And one would think that, well, how does the mechanical properties of these materials change? The cellular solids modeling suggests that the relationship between modulus, the modulus of this material depends only on the relative density, not on the pore size. And that's in fact what we find. So that as we can change the pore size of the scaffold and keep a constant modulus of the material because we've kept the relative density constant, or we can keep a single biomaterial and actually change the cross-linking density to change the mechanical properties. So this allows us to go back and forth between changing microstructural features and mechanical features independently of another. And that's sort of the basis for a lot of the work we started to do. So there's other ways in which you can integrate cellular solids modeling theory into your biomaterials. Another one is to look at the relative amount of isotropy or anisotropy of your material, depending on whether you have a relatively isotropic unit cell versus a highly anisotropic unit cell. The relationship in elastic modulus in orthogonal directions would be different. And so we found that for isotropic materials we make, there's no difference in elastic moduli across all three axes. And some anisotropic materials we make, there's a very significant difference. And it's one that, again, that you can predict. So what I'm trying to harp on here is that we're trying to build a biomaterial system within the concept of actually having a modeling framework that can describe it. All right, so one last step is we wanted to get back to is we wanted to validate again this relationship I talked a little bit about, where you can relate the elastic modulus of your biomaterial to the elastic modulus of the material that it's made out of and the relative density squared. So what that suggests is that we should be able to perform mechanical tests on an individual fiber from the scaffold and mechanical tests on the entire scaffold itself and actually relate them together. And we've done some work there where we've done macroscopic compression and tension tests, then actually cut out individual fibers and done AFM tests on them to actually do bending tests. And we've actually related quite well to find the elastic modulus of the dry and hydrated fibers on their own. They're up in the megapascal ranges, and actually see a nice correlation between our measured elastic moduli of the fiber, our predicted modulus of the entire scaffold, about 30 kilopascals, 27 kilopascals, and the modulus of the scaffold, this one variant I'm describing was actually 30 kilopascals. So we're seeing really good correlation between modeling predictions and experimental results. And so this is starting to give us the basis, we think, to have a series of materials we can use for a number of quantitative measurements of biology. One last thing to give you a sense of is that cellular solids theory is not just useful for mechanics, although it's kind of one of the more initial things we looked at. You can actually develop model theory to describe a number of physiological properties of a porous material. Another one that you as a tissue engineer might care about would be the permeability of your material, so the ability to get nutrients into your scaffold. And so you can actually, and I'm not going to go through the model formulation, but you can actually develop a relationship to describe the fluid mobility or permeability. It's just that there's a different constant that goes out front of your biomaterial in terms of the pore size of the biomaterial, the applied strain on the biomaterial, so if you're loading in a bioreactor or putting it into the body, and the relative density of the scaffold you start out with. And so what we have here is a rather complicated chart, but what you see is solid and striped bars. The solid bars are actually experimental measurement of the permeability of four of our scaffold variants with pore sizes between 95 and 150 microns under 0, 14, 28, and 42% strain. And then the striped bars is the model prediction that we have with one constant up the front. So there's only one constant that actually describes the behavior of all these biomaterials together. So what it allows us, again, what I'm trying to give you a sense of is that you don't have to just make a biomaterial and give a good guess at characterizing it and move on. You can actually do a lot of really careful modeling and characterization work to give you a much better sense of the environment that you're creating. All right, so let's now start talking about using these materials. So we've made a series of biomaterials where we can change features such as pore size, relative density, which I didn't talk too much about, mechanical properties, all independent of one another. We want to actually use them as model systems to study some cell behaviors that we might care about. And so the two that we've done some work in the past actually relates to cell migration and contraction. Again, this goes back to the tissue engineering basis for a lot of this work. The concept being that cell-mediated contraction is very significant in terms of a physiological response to injury, particularly severe injuries. So what this an image up here is actually it's a 12-year-old boy who had burned his chest with boiling water and was treated with a conventional allograft material and with a collagen-based biomaterial that had been developed by one of my doctoral mentors about 20 years ago. And what this biomaterial had been designed to do is actually prevent cell-mediated contraction. And so what you saw is that when you looked at the healing a year later, the area treated with a conventional skin autograft had a lot of associated contraction and scar formation, and the area treated by the biomaterial designed specifically to prevent cell contraction actually led to regeneration of a fairly complete skin. There was no sweat glands, no hair follicles, but functionally it actually behaved like skin, mechanically it was like skin, and aesthetically it was like skin. So that's one area that we're very much interested in. If we're trying to design biomaterials to affect the cell-mediated contraction events we see in native wound healing, it would be a good idea if we could actually use these same material structures to understand how cell contraction takes place. And similarly, cell migration, which people care about for a whole host of reasons, from anything from embryonic developments, understanding tumor cell migration, a number of wound healing processes. Even just if you're designing a biomaterial, one of the key things you want to understand is how quickly you can get cells to move into your biomaterial. And so what we've been trying to develop is techniques you can measure contractile and migratory behavior of individual cells. Traditionally, this has been done by the field using 2D substrates, and we've been trying to look at ways in which we can use our three-dimensional collagen materials and our underlying modeling framework to start to look at similar types of behaviors. So what do we know about cell motility? This is more of a background of some of the processes that are understood. You know, there's a number of major phases in cell migration in terms of membrane perfusion, cell contraction, detachment. And what's not clear is, how these processes are affected as you go from 2D to 3D environments. So the 2D environment is one which a number of groups have looked at cell motility several times. One of the classic examples is Julie Wang's group, who was at UMass when we did this with Harley Mellon. And they were looking at using polychromide systems with tuned mechanical properties to look at fibroblast migration. So not only did the underlying mechanical properties of the membrane affect a number of other features such as traction forces, DNA synthesis, and apoptosis, it also affected cell migration. And they could watch cells on an interface between a soft and a stiff biomaterial, where the difference was only about 14 to 20 kilopascals, which is not something you and I can really feel. But what they could see is you could see quite substantially cells would move quite happily from the soft onto the stiff side of the membrane. But then when they turned around and came back the other direction, what you saw is that the cell would come to the membrane, make a decision, and not cross over onto the stiff side, and would stay onto the soft side, and would stay on the stiff side of the membrane. So what this is starting to suggest, and it's been work that's been validated and built on in a number of different systems and a number of different cell types, is that there's a great deal to understand about the local mechanical properties, and as we're starting to look at local microstructural properties and how that affects cell behavior. So what we had now is a system that we had built that we could actually affect scaffold modulus and the mean pore size of our scaffold independently of one another. So we thought this might be a really neat tool and a neat model system to start to look at similar types of behaviors that Julie Wang's group and a number of other groups looked at before, to try to get an understanding of features such as cell migration and cell-mediated contraction, and is there a complex relationship between pore size and microstructure and mechanical properties that you don't necessarily get out of a 2D membrane system. And in fact, you can look at lots of other features as well, not just pore size and modulus, vector cell density, cell type. There's lots of studies that can do that. And so what we found was a couple of interesting things. First of which we look at, what you see here is a confocal microscope image of cells staying green inside of a scaffold staying red. And so what you see is that it's this nice three-dimensional structure. Cells are sitting on the struts and they're working their way through the different fibers of the scaffold. And in fact, one of the first things we noticed is, again, a cellular-solids relationship, is that when you looked at plotting the cell attachment at 24 and 48 hours within these scaffolds against the calculated specific surface area of our scaffold, which is the amount of surface area available for cells to bind onto, we found these beautiful linear relationships. And again, this comes out of the idea of modeling theory that allow you to make predictions about how much surface area is available for the cells to attach to based off of the microstructure of the scaffold you're creating. But what was actually a little bit more interesting is that we started to change the pore size of the scaffolds what we saw is that this is a windrows plot down here. And the idea is that we took, this was 50 cell migration tracks from inside some of these scaffolds with pore sizes between 95 and 150 microns. And we followed these tracks over 10 hours and represented them by starting them all at the origin. So what you see is when you see it, like for example, this scaffold right here, you see lots of tracks going quite far away from the origin. And this plot over here has not as many tracks and they're not going nearly as far away from the origin. The scale on all these are the same. So what this is meant to represent is that there not only were there differences in cell attachment as we changed the pore size of the scaffold, but there were huge differences in the motile behavior of these cells. And it wasn't just the total distance that cells moved. We saw relationships about the relationship between the strut stiffness of our scaffold. We saw sort of this biphasic peak where on soft materials the cells were sort of migrating about 10, in this case 10 microns an hour. As you increase the stiffness, it almost went up to 15 to 16 microns per hour and then went back down again as you went to high stiffness biomaterials. We also saw that independent of scaffold stiffness, as we changed the microstructure of our scaffold from these small pores of 95 microns all the way up to 150 microns, we saw, again, significant differences in cell motility. And so this got us to start thinking, is that this type of relationship is actually not that surprising, understanding the mechanisms of cell migration. But this one is actually quite a bit surprising, is that we're thinking about, you have a cell that might be 20 or 30 microns in size, and it's sitting inside of a pore that's 95 microns or 150 microns. You know, so we're talking about a cell inside a big house or a bigger house. So why is it that there's any sort of behavioral differences that we're noticing between these scaffolds? And what we found is that having the modeling framework and these sort of standardized series of cellular materials allowed us to start making mechanistic predictions of what's going on. So what I mean by that is we looked at a number of different properties, and I won't talk about all the ones that didn't work. I'm going to talk about the one that ended up being what we concluded at the end. Is when you look at sort of the generalized cellular material that is the scaffold and the cell within it, and this is not drawn exactly to scale, but to give you an idea of what we're looking at here. So you look at an individual cell within the structure, what you see is the cell, they don't float in the pores, they actually stay bound onto the scaffold structures. So what you see is these cells go from being stretched along almost 1D fibers to when they get to these junction points, they stretch out in sort of 2 and 3-dimensional conformations. So there's very significant changes in the cytoskeletal organization of these cells and the mechanical and microstructural features exposed to the cells as they move along through our materials. So this allowed us to start to make some predictions and then go back and make some experimental measurements about what is going on. So you can use your cellular solids theory if you really wanted to get a sense of how does the spacing between junction points change as pore size changes, or what's the overall density of junction points within a biomaterial. Again, it relates to the average diameter of the scaffold, or the inversely proportional to the average diameter cube of the scaffold, depending on whether it's the spacing or the density. And so what this allowed us to start thinking about is making a prediction of what we're seeing was actually we were seeing this sort of persistent migration along the fiber struts, some sort of turning or decision-making process that's happening at the junction points, and then followed by more persistent migration. So we could actually go back into our data and actually look at the cells and break it out into when the cells are moving along the fibers versus when the cells are moving at junction points. And saw that not only were there huge differences in migration along the fibers due to pore size, but also there was significant differences in the migration of cells at the junction points that also scaled with the pore size of the scaffold. So what this allowed us to start to say is that, you know, we don't have this totally nailed down yet, but at least having a modeling framework and a homologous series of standardized materials gives us a running shot. It's starting to make quantitative measurements of exactly what is going on from a mechanistic perspective, how cells behave inside 3-dimensional materials. So onto another sort of biosensing application is cell contraction events. And so there's 2 types of cell contraction events that we've started to look at, one of which is sort of a macroscopic event, which is, you know, you take 1 million or 2 million or 10 million cells, you put it in a biomaterial, you measure the overall dimensional change in the biomaterial over time, and you can make an estimation of how much force the average cell is generating within that. There's a number of assumptions you have to make there, but then there's another approach that we started to look at, again, based on this idea that we now know the local microstructural and mechanical properties of our scaffolds and have a model and framework to describe it. We can actually use in this case, this is just a light microscope looking inside our scaffold structure, and that's a single dermal fibroblast. It's sitting on a strut of our scaffold. Here's some other struts. And we look at it over time, and we can actually watch it contract and buckle the strut it's attached onto, and it not only buckles the strut, it ends up deforming the entire local microstructure of the scaffold. And this is something we've seen for a very long time when we looked at histology images of when we had seeded cells in our scaffold and came back 3 weeks later. You could see significant changes in the microstructure of the scaffold, but we can watch it in real time now. And so what this allows us to do is that, well, we can make 2 different types of measurements. And so the first measurement we've been comparing it to was this sort of cell-mediated contraction of populations. And again, this idea is we take our scaffold, we seed it with 1 million or 2 million or 10 million cells, put it into some sort of measuring device. In this case, it was a colleague of mine, Toby Fryman, built the cell force monitor approach, which basically attached the scaffold to a very flexible beam and then measured the deformation of the beam as a way in which you can measure contraction of the scaffold. And he could generate these really beautiful curves of looking at the time constants of the average contraction force generated within these cells. So there's a time constant of about 5 or 6 hours in this nice sort of exponential curve, actually an asymptotic curve, where they could actually look at the average force being generated up about 1 to 2 nanonewtons per cell. But the other interesting thing is they measured at the exact same time the aspect ratio of the cells. So what they could measure is they could see cells within the scaffold are going from being rounded after being seeded. They're becoming increasingly elongated, expressing higher amounts of alpha smooth muscle actin, and actually having a much more structured cytoskeletal organization than the cells right after originally seeded. So what they saw is they could relate macroscopic changes in the deformation of the scaffold and cell-mediated contraction to actually changes in the local microenvironment of cells. And they could look at the effect of a certain number of cells, or they could actually look at the effect of the stiffness of the system. And what they saw is that when they put these cells into different scaffolds with different stiffnesses associated with them, cells didn't generate different amounts of force. They generated a single amount of force, but you saw different amounts of deformation in the biomaterial. So it sort of makes sense that if a cell is going to be sensing, the mechanical environment around itself, it's not necessarily a huge, complex stress strain measurement it's making. It's going to apply some known amount of stress and going to measure some underlying buckling or deformation, and that's the way in which you can start to make measurements of mechanical properties. But on to what I think is kind of a more interesting application of sort of biosensing is if you actually want to make measurements of individual cell contraction events. And so this goes back to the video I showed a couple slides ago, where you had a single cell sitting in a fiber within the scaffold, and over time the cell contracted and the fiber buckled. And so there's actually, again, there's a modeling framework you could use. It's based off of sort of modified Euler buckling equation that has a prefactor out the front that's actually associated with the cellular material itself. So what you're seeing here is you're having an individual fiber that's buckling, but it's not like it's pinned at either edge. You have either end of the fiber is actually attached to the much larger scaffold structure surrounding it, so it's almost a rotational stiffness that's associated with the scaffolds is the way you model it. And so what you can do is you can actually generate measurements of contractile force generated by individual cells based off of the modulus of the strut of the scaffold, which we've measured experimentally and confirmed using a modeling theory, and a prefactor that's based off of the type of contraction you're seeing, whether it's single direction, multi-direction, or more of a hydrostatic type contraction. And we can actually use this to make calculations based off of looking at individual cells that started as rounded cells on an individual fiber, and you come back an hour and 40 minutes later and you see the strut is quite buckled. You can make calculations that we measured a number of different cell scaffold systems, and we found a fairly conserved contractile force, about 25 nanonewtons per cell was being generated. We didn't see forces generated lower than 11 nanonewtons or forces generated higher than 41 nanonewtons. And in fact, this is something that there's been a few other groups that have started taking and run with and have designed quite frankly, better fiber systems, 1D fiber systems to look at, and they're finding very much consistent numbers that we're finding. And so it's kind of been fun to think that we can make individual cell measurements based off of a modeling theory and experiments itself. In fact, one of the other things you can do is, one of the things we noticed is that there were sometimes where cells couldn't buckle the scaffold. So we'd see some cells into a highly anisotropic scaffold that had some really thick, high-density structures within them. And what we saw is there's an individual cell that started rounded here. And over time, it started to elongate. And it started to generate some sort of tension within the cell, and that's the point where the focal adhesions actually sheared off and the cell detached from one side. And what we watched it do over the next couple hours is actually round it up, and it started to then elongate again, and it again generated this nice high aspect ratio structure, and very, very soon that it broke again. And so what we could watch is we could watch an individual cell inside this 3D scaffold structure attempting to contract a fiber, but not actually being able to generate enough force. But because we were able to measure the local geometry of the strut structures. We could actually make a calculation of how much force it would have taken to buckle that strut region. And this one worked out to be about 450 nanometers. So again, this is an idea of ways in which you can combine modeling and experimental results to actually make some interesting measurements of cell behavior. All right, so now that we've started to develop a modeling system where we have a homologous series of biomaterials, we have an underlying framework of sort of modeling approaches, we've been trying, and you'll have to excuse me, this is actually the first time I've really tried to keep this part of the lecture before. It's still it's sort of an open concept we're working on in my group. It's this idea of unit cell processes that has actually been introduced by Myron Spector, who's at the VA Hospital Boston and teaches at MIT a little bit a few years ago. The idea is to try to develop some constituent framework for tissue engineering to understand cell processes in more of a greater context. So the approach is you define a control volume surrounding a single cell, and then you come up with almost a chemical reaction relationship to describe some process that's taking place within that cell. And so we think this, and I'll explain the concept on the next slide, but we think this unit cell process, or UCP concept, actually will allow us to provide some greater insights into the relationship between a number of cell processes. So if you have multiple cell processes that take place together, such as mitosis and synthesis, and it might relate to contraction, or how does migration relate to contraction. This might allow us to provide more insights into cell behavior in vivo, but in general, it allows us to actually serve as a model system for us to design new experiments to try to actually come to a better understanding of how cells behave in a three-dimensional environment. So here's the concept behind it. You have some sort of cell, and it's acting in contact with some sort of matrix, and then you have, so these are the reactants, and then you have a product. And there's lots of different types of products that might take place, and there might be some soluble regulators that cross the control volume to affect the cells early on. There might be some soluble factors that leave as part of the product that could go out and affect other cells. There's ways in which mechanical force could actually be directly affecting onto a cell, where you're deforming a cell due to some sort of loading or fluid flow, or the fact that mechanical force deforms the matrix, which indirectly affects the cell. There's lots of ways in which you can start to describe this. So there's lots of types of cells you can put into this, cells that it could be based on connective tissue, epithelial, muscle, nerve. We would try to build this framework up. And what is a UCP? Well, there's lots of UCPs. It could be mitosis, synthesis, migration, contraction, quiescence, self-renewal, differentiation. The idea is to try to think about a minimum element of the cell behavior and build it up from there. So for example, if you think about bone remodeling, we know it's sort of an interrelated process where you have osteoclasts and osteoblasts, where osteoclasts actually synthesize collagen, collagenase, and in order to solubilize the local bone matrix, this process also releases some soluble regulators like TGF-beta, which actually upregulates osteoblasts to produce new matrix. And this process actually releases some soluble factors, prostaglandins, which can actually induce osteoclasts to upregulate their behavior again. And there's mechanical forces that take place because we know that mechanical forces can actually stimulate bone formation, or a loss of applied mechanical forces can result in loss of bone, such as space flight, or that's a classic example. So this is the idea. You start to develop this unit cell process where you have a cell, some sort of matrix, and a resultant product, and there's ways in which you can interrelate them. And the goal is that we can actually start to not just have these qualitative images, but actually, because we can make measurements and define biomaterial systems, actually start to put quantities, so you have to add in reaction kinetics and stoichiometries in terms of ways in which we can describe cell behaviors. So what are lots of different processes, and just I figured I'd throw a few out there. So for example, the one process is mitosis, and the product is more cells. Another one might be exocytosis, and the product is lots of different regulators or those biomolecular factors that are released outside of the cell. Contraction could be a process, with the product being some sort of resultant stress or strain on your exercise matrix. So there's lots. So just to give you an idea of how this could start as a qualitative process, where you could have, clotting is a process where you have platelets acting on collagen, that they produce this degranulation process, and it results in platelet aggregation within the local environment. And so this is just a way to start putting together a qualitative description of what's happening inside of our biomaterials. And the idea behind the quantitative comes from that there's lots of folks out in the literature, and we're starting to do more of this. We can actually look at fibroblast mitosis, for example, and look at the effect of PDGF or FGF, TGF-beta, IL-1, TNF-alpha, all these different factors, and get a sense of whether it has a positive or negative, and hopefully actually start to get more quantitative relationships between concentrations of factors and measured behaviors. The goal being that you can start to build up large numbers of composite or supercomposite unit cell processes. So one doesn't necessarily care a lot about collagen degradation on its own, but you want to understand collagen remodeling as a process that has collagen degradation and collagen synthesis happening together and an interrelationship between these processes. So by starting to have a sense of the stoichiometry and the reaction kinetics associated with that process, we might be able to start to design better materials to manipulate the behavior or take advantage of that behavior for tissue engineering applications. So these are just examples. Composites are two single unit cell processes together. The idea of a supercomposite is you have multiple composite UCPs together, where you have tissue remodeling has collagen remodeling, neovascularization, scar tissue formation, a lot of different complex features, but by breaking them into individual elements and putting together a larger structure, you might have a chance of being able to understand these processes in a larger context. So this is just like the concept of how do you understand response to implanting a biomaterial, where you might have surgical implantation of material into a wound site. You've got all sorts of acute inflammatory processes, vascular response, clotting, phagocytosis, neovascularization, collagen synthesis. Some of this is also the formation of granulation tissue. And depending on the material you put in there, you can sometimes see this idea of the formation of a tissue of permanent cells. And so this is actually due to scar tissue formation. You have chronic inflammation that's taking place. Or you could actually generate some sort of label stable remodeling tissue that could actually result in regeneration if you do it right. So starting to understand the relationship between lots of different factors within the granular scheme might give us a chance of understanding what's happening. So this is just the idea of complex unit cell processes, vascular response to injury. If you start to think about it, you have platelets, basophils, and mast cells are all, there's a lot of exocytosis taking place that can stimulate endothelial cells to start local leakage, and eventually will actually start stimulate smooth muscle cells to lead to vascular constriction. So there's a really complicated process that you can think about. You can think about other things like just the clotting process itself. But the idea in the longest idea is trying to take advances that we've been seeing in tissue engineering biophysics to allow these unit cell processes to become quantitative and be able to start to understand the reaction kinetics and stoichiometry. And a lot of this is founded on the idea of doing these types of experiments in a longer series of materials, because that gives you the framework to understand quantitatively what's going on. Not perfectly yet. We're nowhere near perfect. I don't want to suggest that. But at least we think we have a direction we're taking which we can start to ask questions from a mechanistic standpoint. All right, so now on to some more fun stuff that is a little less mechanistic, and I just want to put it in at the end, is actually starting to, the other application of our biomaterials isn't just to do some modeling work with it and understand cell behaviors, but can we actually use these to do something useful from a tissue engineering standpoint? And so my group has worked quite a great deal in developing materials for orthopedic tissue engineering applications. Most of the background of these collagen scaffolds, they were developed for soft tissue applications. They were developed in the early 70s, have been used for skin and peripheral nerve regeneration clinically so far. And that was where I came up as a grad student, and so I had to go in a different direction rather than just doing that again. And so decided we try to take these to orthopedic materials. One of the catches we found is that these collagen scaffolds are very soft. They're good for soft tissue models, but not that great for orthopedics. So we had to come up with a lot of new fabrication approaches and concepts in order to build tissues that are actually stable and able to be used in orthopedic applications. So this comes back to the idea of understanding mammalian wound healing, so the contraction and migration events, and that the historical background of the work that I do is that there's this intricate process following injury where you have this initial inflammatory response, proliferation and remodeling. What has been noticed by a number of different investigators that there's a transition as you go from fetal development to adults after you're born, is that in the first 2 trimesters, most mammals have the ability to regenerate a lot of damaged tissue. And there's some transition that takes place. Some of it has to do with some TGF-beta signals. There's a lot of other groups trying to understand a lot more detail about the developmental aspect of this. But you and I get to the point where you and I can get a paper cut, and you come back 2 weeks later and you don't see anything because it's just a very small volume of a cut. Whereas if you get a severe burn, or I've got scars on my fingers from when I was a kid, scraping them when I'm riding my bike or something like that, you can, once you get to a large enough wound site, you actually see this formation of scar tissue and cell-mediated contraction. And so what we're trying to understand is how can we actually produce a material to implant into this defect site to simulate the local extracellular matrix environment well enough that it leads to a remodeling and regeneration event, prevents the organized wound contraction and scar tissue synthesis you would see if you had an ungrafted one. And so one of the areas we've worked in is bone tissue engineering. So the nice thing about bone is that we think of trabecular bone. It's this nice low-density open cell foam. Granted, it's a mineralized low-density open cell foam, but we've worked a great deal in actually developing fabrication processes to produce collagen-based scaffolds that have a collagen backbone with an integrated nanocrystallized calcium phosphate to mimic the local structure you see of bone itself. And it's done with this triple-phase co-synthesis process where we take our collagen and our glycinated glycan and calcium salts and a phosphoric acid, and by titrating the amounts correct, you can actually precipitate out nanocrystalline calcium phosphate as the collagen that you're precipitating out of solution during the fabrication. Then you go through your freeze drying step to set the pore size and shape and all that sort of fun stuff. We've ended up coming up with this three-dimensional mineralized scaffold. It's got a relative density of about 15%, so it's about 85% porous. It's one of the more highly dense scaffolds you produce. And it's produced with a brushite phase of calcium phosphate, and we can use hydrolysis steps to change it from brushite to octocalcium phosphate to apatite, which are a lot of different features of a biomaterial that we might care about. The reason for that is that, going back to a developmental standpoint, if you look at how bone comes to be, you have a sort of this ossification event that takes place, and you have changes in the amount of mineral and the phase of mineral. And so what we've been able to do is make some biomaterials so we can actually have the scaffold represent mineral stages at different points of the development process, to try to get an understanding of, is there a more appropriate mineral phase and mineral content that allows rapid cell integration, rapid remodeling, as opposed to just putting a material that mimics material, which never can produce huge amounts of bone anymore. So that's sort of one of the fun things we've been messing with. Something else we've been messing with is how do you start to produce complex, multifunctional materials. And bone's kind of fun, but from a biomaterial standpoint, it's, for the most part, it's a little boring. It's a three-dimensional structure. It's quite uniform. There's some local heterogeneities, but from a biomaterials fabrication standpoint, osteochondral injuries were much more interesting. So this is an example of cartilage in the underlying bone. Oftentimes you see damage to both. So the solution for fixing cartilage right now, there's a lot of different clinical solutions out there that aren't that great. But one thing that groups try to do is try to put some sort of gel or cell construct into this wound site of cartilage, not doing anything under the underlying bone, and it's very hard to actually get any sort of material to stick into just the cartilage region because you can't suture it into the surrounding cartilage without damaging the cartilage. There's nothing that really forces the surrounding cartilage to grow into the new material because it's such a low mitotic material. But the interesting thing from the biomaterials design is how osteochondral tissues are developed in the body. And what you have is you don't have cartilage sitting on top of bone with a nice hard interface in between. What you actually see is you have your articular cartilage region, then you have this tide mark region, which is, you have a gradual transition from non-mineralized type 2 collagen to mineralized type 1 collagen bone over this interfacial region and a continuity of fibers across it. So what this means is you have cartilage, bone, and a continuous interface. So what happens is when you're loading your cartilage, if it had a discontinuous interface, so you had just cartilage sitting on top of bone with no interface in between the two, every time you're loading on it, the modulus mismatch between cartilage and bone, you'd see a lot of stress concentrations right at the interface, and that's one of the biggest problems with biomaterials right now is that if you have a material like that and you start loading it, you see lots of delamination that takes place exactly with that interface. So what we tried to do is there a way we could actually make a single biomaterial that had a cartilage region, a bone region, and an interface? That seems like a much more interesting thing to spend our time doing. We had this concept we came up with called liquid phase co-synthesis. Basically, and I'm always somewhat embarrassed admitting where it came up, but I tell it because it's a good story. It came up going to a bar called the Miracle of Science, where Andrew, Lynn, and I, who were the two grad students working on this project initially, were sitting down and trying to think about ways in which we could make this material. And the person next to us had ordered a black and tan, which I don't know if you know what a black and tan is, but it's layering of Guinness and stout alcohols. And so if the bartender's good at their job, they can actually layer these two alcohols and keep those two alcohols separate within the glass, except for a little intermixing region right between the two, where you actually see a lot of foaming and formation of this interface. And so we thought, hey, that'd be kind of fun if we could actually take our liquid phase materials, our type 2 collagen suspension, our type 1 mineralized collagen suspension, come up with some interdiffusion conditions in terms of time and temperature to actually get the two suspensions to interfuse a little bit, then go through our freeze drying process, and maybe we'll get something. So a few years later, we actually came up with this a scaffold that has this is a micro CT image of the side of our collagen scaffold that has a highly porous type 2 collagen gag scaffold, cartilaginous compartment, and a mineralized type 1 collagen osseous compartment, and a continuous interfacing between the two. This is a SEM just looking at the interface where those collagen fibers going across, and this is an EDX image showing calcium phosphate concentration in the scaffold. So we have in red superposed with the scaffold. So we have non-mineralized, highly mineralized, and this interface that takes place in between the two. And this is a material that's now being used in knee cartilage surgeries. It's actually in phase 1 clinical trials in Europe. It's gone through the entire regulatory process, and it's something that we're still building on, but it's actually kind of a fun project that we can make not just a single uniform material, but start thinking about producing anisotropic or complex materials. So what's the next material we're working on? Again, this is trying to build cellular materials and starting to use some of our understanding of a modeling framework and producing and the production of these materials to come up with some new structures. And so we started to think about tendons. So tendon in and of itself is interesting, and there's a tendon to bone interface we're also working on. But tendon itself, and lots of different types of injuries, but tendon is a highly aligned and highly anisotropic structure. So all the fibers are aligned in the direction of the tendon, making it a much more interesting project to work on. And there's not a lot of good clinical treatments for tendon and tendon bone insertion injuries. And so the tendon bone insertion is similar to the osteochondral model, kind of an interesting one, because you go from highly aligned collagen structure tendon to this interfacial region. We start to see more and more calcium phosphate mineral, less and less alignment, and sort of a high proteoglycan content right at the interface. Then you end up with an isotropic, highly mineralized bone structure. So again, can we actually make these types of structures with our materials? And so our concept is, can we actually create a multi-phase scaffold for tendon to tendon insertion, where you have a mineralized region, continuous interface-oriented tendinous compartment. And we think there's a lot of things that we can do from the cellular solids and modeling and production side of things to start designing these materials. So we know we need to have some sort of aligned microstructure in the tendinous compartment, has to be mechanically competent, and we're working on adding in biomolecules, which I'm not going to talk about here because I'm going to relate to the class, but not only do we want to have microstructure mechanics, we actually want to have some sort of chemical composition effects as well. So we can start to make highly aligned scaffold structures just by doping the freeze drying process with this idea of directional freeze drying, which is if you take your suspension and you put it in a composite mold that has a high degree of thermal mismatch between regions of the mold, like having a copper base on a Teflon mold. The degree of thermal conductivity mismatch is about 1600. So you start solidifying this, you get really elongated ice crystalline form within it. And these are scaffolds fabricated at different temperatures. And as you get to colder and colder temperatures, the pore size shrinks. But what you see conserved in the longitudinal direction, which are these phases down here, is a highly aligned structure. It's got an aspect ratio of about 1.6 to 1 in these pores. So they're really highly aligned pores. It's a whole aligned structure, and it's one we could produce in sort of a composite. Now, the most interesting aspect, and where we went back to this idea of understanding relationships between microstructure and mechanical properties, is how you design materials to be strong in tension when they're porous. And there's the classic problem that tissue engineering has had, where mechanics and porosity are oftentimes very much related. We talked a little bit about the modeling framework, where the modules of the scaffold scales is a relative density squared. So as the scaffold becomes more and more porous, it becomes less and less stiff. However, if you start making the scaffold more and more dense in order to increase its strength, you actually reduce things such as permeability. So the cells aren't metabolically supported anymore and they start to die. So there's this complex relationship you need to understand. And so there's been a lot of groups working in orthopedic tissue engineering that have gone the route of electrostrong polymer maps. Those work really well in the sense that these high density polymers that can be either isotropic or highly anisotropic, they're very high strength, but cells don't really penetrate into them. They're not a 3D structure, they're a 2D structure. We started to think about nature actually has a solution to all this, and it goes back to the cellular concept. That if you look at a lot of natural materials, there's this idea of a porous shell composite, where you need a porous material that has to be stiff in tension, compression, or bending. For example, a porcupine quill has a really high density sheath around the outside and a very highly porous central core. You see the same thing in bird beaks. Engineers have taken this, and this is a high strength, porous copper conduit that's used for some structural applications. And again, it's linking together high density, high strength membranes with highly porous materials. So we thought, well, it'd be kind of fun to actually see if there's a framework we can do to produce our highly aligned scaffolds with a membrane shell around the outside that actually produces porous shell composites. So we've actually gone about making these membranes where they have a relative density of about 75%, so they're very, very dense, they're very, very stiff. We can change features such as the thickness of these membranes without changing relative density via some fabrication techniques. We can actually start making subpore shell composites. So this is the classic one with the shell around the outside of the porous core. We've actually also started to make some concept of a central core membrane or a partial shell membrane as well. The interesting thing about it is that it actually gives us some really high strength applications. So let's look at some properties here. So if you look at the membrane itself, it has a modulus of about 45 megapascals, which is much higher than anything we see in a scaffold. For example, a classic scaffold might be in the kilopascals if you're lucky, and that's nowhere near the 150 megapascals you see in tendon itself. But when you start to actually take our line scaffold, we take our membrane, put them together, we can actually create a core shell composite. So we take the 200 kilopascal modulus scaffold and actually turn it into about a 1.5, 1.6 megapascal modulus composite in the end. So we're working on ways to increase the density, increase the size of the membrane, and mess around with a few other things. But there's actually a really nice modeling system we could use. We have our cellular solids modeling to give us a sense how the scaffold structure change, how the scaffold mechanics change as we affect relative density or pore size or cell alignment, those sorts of things. And then there's the idea of layered composite theory, which actually does a very good job of predicting if we have a membrane of a certain dimension and stiffness and a scaffold of a certain dimension and stiffness, it can actually predict what our composite will be quite accurately. So again, it gives us the idea that we can produce a series of these scaffolds to validate a number of modeling tools and actually use the modeling tools from a design standpoint to say, if we need a composite to have a certain level of mechanical stiffness, how do we actually build it? So that's about it. I just wanted to give you the last couple of fun things that we're working on just as sort of an end here, is that not all of this has to be, you know, super mechanistic. You could have some fun in producing biomaterials for sort of biosensing applications. So what we've been trying to do is, in the last couple of slides I show you, how do we produce these next generation of spatially temporally patterns in biomaterials that have gradients, multiple compartments, pattern heterogeneities that could be destructive. So that's what we're working on now, is that we're trying to think if there's ways we can actually make these patterns constructive. And so this is something that's the biomolecular aspect I didn't want to go into very much, but we can actually take our collagen egg scaffolds and use a benzophenone photochemistry approach. We're working on this with a colleague of ours here at Biomolecular Chemistry, where we take our scaffold with defined microstructure and mechanical properties, and we can attach onto the scaffold through the free means on the collagen surface this benzophenone molecule. And the interesting thing about benzophenone molecule is that when you irradiate it at 365 nanometers, it forms this transient diradical, which will allow insertion of any CH bond. So you have some sort of biomolecule floating inside of your scaffold, you hit it with UV light, and it attaches in the local region where the UV light is exposed, whatever biomolecule that is. So we're just starting to mess with this, but we're doing about 350 molecules per micron squared, so it actually gives us the ability to pattern locally at the resolution of UV light, or starting out to use some two-photon microscopy approaches to do it, pattern structures into our scaffold. So this is a uniform isotropic scaffold that we've now patterned stripes of, in this case it's just a quantum dot for imaging purposes, all the way through the scaffold. Or it can do bidirectional gradients of n-catenaries to look at cell mediated attachment. So again, this goes back to how do you start to produce suites of materials to allow you to actually use the constitutive modeling to make measurements of cell behavior. So the last thing is just that these patterns are limited only really by our imagination. So our fun is that you take the Illinois logo, and the next thing you know, we have the Illinois eye pattern into the middle of one of our scaffolds. And so there's lots of fun things you can do in biomaterials as well. So I'm just going to skip this, just because it's at the end. And I want to thank you all for your attention and say that none of this work would have been possible without the really hard work of all of my grad students and undergrads, my collaborators on campus and throughout the world, and the funding we have to support this work. So I'd be happy to, in the next few minutes, answer some questions or just be around for lunch to talk about things.